Good morning or afternoon to you. Thank you for joining today's webinar. I'm Jan Reichert, Executive Director of the Antibody Society, and I will be your moderator. This webinar is the second in a series designed to inform and educate our members and the broader scientific community about antibody discovery and development. The title of today's webinar is Ultra-Fast Development of an Anti-COVID-19 Antibody. Our speaker is Dr. Andre Frenzel, who is the Chief Scientific Officer at UMAB as well as Corat Therapeutics. He's been involved in the many aspects associated with this very intensive hunt for anti-SARS-CoV-2 antibodies that can be used to prevent or treat COVID-19, and we'll hear about his adventures today. This webinar is being recorded. We'll take questions at the end of the presentation, so please add your questions to the Q&A box in the viewer. Without further ado, I'll turn the show over to Dr. Frenzel.
Thank you, Janice, for the kind introduction, and thank you also for inviting me to this very interesting webinar. And you mentioned already, this is really a hunt for a treatment against COVID-19. And as we are talking today about ultra-fast development of an antibody, anti-COVID-19 antibody, I'm not talking to you in a compressed way, as is usual for such kind of scientific talks. But I'm trying to tell you the whole story in the timeline as it occurred. Just a very brief introduction into the infection cycle of SARS-CoV-2. I think we all know the fellow who's, let's say, who's our companion over the last 12 months, I think. Just to give a very short introduction about how SARS-CoV-2 is infecting. It is binding to the ACE2 receptor on the cell surface of the host cells and gets activated by a protease named TMPRSS2, which cleaves the S1 protein of the SARS-CoV-2 particle, and therefore the fusion peptide is exposed on the surface of the virus. Which leads to the infection of the host cell due to membrane fusion of the virus and the host cell. Antibodies can prevent this kind of uptake into the host cell by blocking this interaction between ACE2 and the SARS-CoV-2 particles here. Because then if the antibody is not able to bind to the cell surface, TMPRSS2 cannot activate S1, and usually this will prevent also the membrane fusion and the infection of the host cell. So neutralizing antibodies can be used to treat sick patients, to treat COVID-19 symptoms, but also to treat healthy symptomless infected patients to prevent an onset of infected patients. But such neutralizing antibodies could also theoretically be used for prophylactic treatment of healthy patients with increased risk of getting a severe SARS-CoV-2 COVID-19 infection. How to get these kind of antibodies? There are several different technologies available, and I think most of you already have heard about those kind of technologies, but I want to give you a very brief introduction into that. You know that or you can do B-cell cloning from infected people to identify antibodies that are able to neutralize SARS-CoV-2, or you can use, for instance, transgenic mice or normal mice, non-transgenic mice. You immunize them with a spike protein, for instance, generate hybridoma or directly screen B cells for neutralizing activity. We use another approach, which is antibody phage display technology, which I think is also very well known in this field here. Just a brief, a very short introduction into that. We can use healthy donors, so a library, antibody gene library from healthy donors with a universal repertoire, which is able to generally provide antibodies against all different kind of antigens. But we can also use the B-cell repertoire from convalescent patients which had undergone a natural infection. Those libraries can be screened for binding to the surface protein of the SARS-CoV-2 particles, and then can be checked for virus neutralization, and can be developed further, let's say in the process development and manufacturing steps, to have at the end of the story a therapeutic agent which could help, which can help people which suffer from a COVID-19 disease. We use antibody phage display, or we used antibody phage display to generate neutralizing antibodies against SARS-CoV-2. And in general, we used both strategies here. We used our large naive antibody gene library, but in parallel, we also had access to material from convalescent patients at the beginning of last year. So we generated so-called immune libraries from those patients, and how it was done I will describe you in the next few slides. So we took the material from those patients which had undergone COVID-19 disease, a moderate COVID-19 disease. We checked the serum of those patients for binders against the receptor binding domain of the S1 protein of SARS-CoV-2, and we only used those sera for generating of the libraries which showed a significant binding to the RBD. These are the graphs here you can which are highlighted in the different colors. In gray, you can see the serum where we do not, which were derived from the patients, which were not included in the libraries, and in the black dot here you see the negative control, which is serum from a healthy donor. We isolated plasma cells using flow cytometry from the peripheral blood of this convalescent patients. So we stained for CD19 and CD138 and isolated those plasma cells, and amplified antibody genes from those plasma cells using specific primers, cloned these antibody genes into the phage mid vectors, and packaged the antibody phage particles, and checked for antibody presentation using Western blot analysis. And you see here this Western blot on the top right side, where you can see the SDFB P3 fusion. P3 is a protein on the surface tip where the SDFB molecule is covalently connected to, so genetically fused to, and on the bottom you see the non-fused P3. Those libraries have then been used to check for binders, or to isolate binders which specifically bind to the receptor binding domain and to the S1 and S1, S2 domains of SARS-CoV-2. So in general, you take these libraries, you incubate it with the immobilized antigen of interest, wash away all the antibody phage particles which do not bind, or which just weakly bind, amplify the bound phage in E. coli, and use those antibody phage particles for the next rounds of enrichment. And after 3 rounds of enrichment, we produce soluble antibodies from thousands of different clones to check in an antigen ELISA for binding to the RBD and to the S1 protein of SARS-CoV-2. And in total, we were able to identify 735 specific antibodies which bound to these surface proteins of SARS-CoV-2. The next step was, of course, to identify the antibodies which were not only able to bind to the surface proteins, but which are also able to somehow inhibit the interaction between ACE2 and the receptor binding domain of the S1 protein. And therefore, we generated, or we set up two different assays. The first one was a cell-based assay. So we transfected ACE2, or we transfected HEX cells with human ACE2, and we fluorescently labeled S1, S2 trimer, or RBD with a fluorescent dye, and we checked for binding of the S1 protein in presence of the different antibodies, which have been converted into an scFv-Fc format, which is an scFv format which is genetically fused to a human Fc part, which can be expressed in mammalian cells and can be purified. So in total, 135 scFv antibodies have been converted and checked in this kind of assay, and we identified 30 clones which inhibited the binding of S1, S2 to the ACE2 presenting HEX cells with more than 80%. So this was the threshold we set over there. In the second track, let's say, of discovery, we set up a high throughput ELISA-based assay where we used an inhibition ELISA to identify antibodies which already have already in the scFv format without reformatting into the scFv-Fc format, which are able to block the interaction between the receptor binding domain and ACE2. And we analyzed 180 scFv antibodies, and from these 180 amongst these 180 scFv antibodies, 137 clones have been shown to inhibit the interaction between the receptor binding domain and the RBD in ELISA. To narrow down the number, because the next step are somehow critical in terms of numbers and in terms of numbers of different antibodies that can be handled in the assays. We clustered those ELISA identified antibodies based on the sequence, and we identified in total 67 different sequence clusters. And in these roughly 70 sequence clusters, 48 clusters included ACE2 RBD inhibiting antibodies, and from each of these clusters, antibodies have been chosen for the next step. For the antibody neutralization assay, so an assay which was set up to identify the antibody neutralizing to identify antibodies which are able to neutralize live virus isolates in cell culture. So we used all antibodies which were able to inhibit the ACE2-S1 interaction in the flow cytometry analysis. Based on let's say different strategies, we narrowed down the number of the antibodies which have been identified in the ELISA-based assays. We first deselected antibodies which have, in our point of view, a so-called no-go criteria, post-translational modifications, which can be predicted or can be seen, such as unpaired cysteines or glycosylation patterns or glycosylations which are predicted in the sequences. In the second step, the remaining clones were selected based on the number of so-called developability flags, which have been bioinformatically predicted. And clones with the identical number of flags were selected based on the ACE2 RBD inhibition assay results. And this last step of narrowing down the number and the clone selection, clones with a similar ACE2 RBD inhibition were selected based on binding characteristics, meaning how good they are binding to the RBD or the S1, S2 protein. So based on these parameters, we selected 44 antibodies for the conversion into IgG and virus neutralization assays, plus an additional 30 scFv-Fc antibodies and IgG antibodies, which have been identified in the flow cytometry analysis. Here's just an idea about the binding assay on the receptor inhibition assay in ELISA, which took place as one of the, let's say, steps to diminish down, to reduce the number of antibodies to take on into the, to move on into the next stage. And you see here that, let's say, the top 3 candidates already have a sub-nanomolar IC50 in this kind of inhibition assay here, where we pre-incubated the RBD with the antibodies, with the IgG antibodies, and checked the binding of the RBD to the ACE2 in presence and without addition of the antibodies. So all these antibodies have been selected for the virus neutralization assay. Here we pre-incubated the SARS-CoV-2 virus with the different antibodies, IgG or scFv-Fc antibodies. They were mixed with virus-6 cells, and this has been used in a plug assay. So you can see here on the top right side 3 microscopic pictures. On the left side, you see the microscopic picture of the uninfected culture. So let's say you see this, that the cells were nicely growing. In the middle, you see an untreated control with a lot of round cells here and a lot of plaques which occurred. And on the right side, you see a microscopic picture with the cells which are treated with a neutralizing antibody. And on the bottom, you see a bar chart of a single point measurement. This is just one concentration which was used here in this high throughput plug assay. And we identified, or let's say not we identified, but we selected 19 clones for determination of the IC50. So in a more an assay which led us to more data, and which give us an idea how good these different antibodies are. The first plug assay took place in 96 well plates. And you see here the dose response curves. So all antibodies have those clear dose response curves and were able to neutralize the SARS-CoV-2 isolate. And the 3 highlighted here with 0.2 and 0.8 nanomolar IC50 values are the top 3 clones, which will, let's say, which will lead us throughout the whole presentation here. After that, we set up a 24-well based assay because we saw that, let's say, the errors get much lower compared to the 96 well plates, because you can see here on the y-axis the number of plugs are much higher in this 24 well assay format compared to the 96 well plate format. And therefore, these data are much more suitable to get real IC50 values here. And you see here the control antibody palivizumab does not have any, as expected, does not have any effect, let's say, or neutralizing effect on the live virus neutralization, whereas the top 3 candidates have IC50 values in the sub-nanomolar range between 0.4 and 0.56 nanomolar. We also checked the binding of those molecules in a direct ELISA, and for those three, for the top three molecules, we have an EC50 which is around, or which is below 200 picomolar for all of the three antibody candidates to the S1, S2 trimer protein, as well as to the RBD protein here. As negative controls, we used here the N-terminal domain of SARS-CoV-2 virus. We checked also, or we determined also the IC50 in the cell-based ACE2 inhibition ELISA of these molecules here, and we could determine that also in this neutralization or inhibition assay, we were able to detect a subnanomolar IC50, which is around 1 nanomolar and a little bit less for all the top three candidates. We were already at a very early stage, let's say in May, June, before the first mutants occurred. I'm curious about different mutations of SARS-CoV-2 virus and how our antibodies behave, let's say, on these mutations. And therefore, we extracted 7 different mutations from literature, which was described at this time, and from the GIS8 collection of different lines of SARS-CoV-2 virus, and we expressed the different S1 variants, including those mutations. And we also included one mutation, the so-called 7PM variant, which includes all 7 mutations in one S1 variant. And we checked for binding of our molecules to these different variants. And you can see here that all variants can be bound by our top 3 molecules. We can also see that all these mutations or variants of the S1 protein are able to bind S2. This is depicted here on the bottom right side of the graphs. Yeah, ACE2 is also able to bind to the 7PM variant without any loss of affinity. This means, of course, we know it, today we know it, SARS-CoV-2 is mutating a lot, and we know that a lot of mutations occurred without any problems for the virus for the infectivity to the human beings. Same has been checked not only in ELISA but also in the inhibition assay. So we checked this in this cell-based inhibition assay on ACE2 expressing hex cells, and we used here fluorescently labeled mutation variants. And you can see here on the top left side, this is the final candidate, let's say, ST90C11, or the new name is Core101. Core101 was able to not only bind to these different variants, but also to block the interaction between these different mutations and the ACE2 on the cell surface here. We then entered a very fast pre-CMC assessment with a set of different analyses we performed here in our lab, in let's say in a low lab scale format. And the first analysis we did here is that we checked binding to so-called sticky antigens. So we used a set of antigens which are known to let's say somehow be a little bit sticky, such as DNA, LPS, lysosome, and also cell lysate, and compared binding of all molecules here with already approved molecules such as trastuzumab, palivizumab, intravenously, immunoglobulins, avelumab, and as negative control VSA. And we could definitely see that there's no sticky behavior of this molecule here. Then we used let's say a treatment with low pH or accelerated temperature to analyze if there's any kind of misbehavior, so aggregation or degradation of those molecules after 24-hour treatment at 45 degrees or at pH 3. And you can nicely see here in the middle, or starting, let's say, in the middle with Core 101 and with the other two candidates of the top three, that we could not see any disadvantaged behavior of those molecules. So they do not have any tendency to aggregate or to degrade under these circumstances here, under this treatment. Affinity measurement was done using biolayer interferometry. We used three different setups here. So we converted the antibodies into a Fab fragment and immobilized the S1 protein and used the Fab fragment as an analyte. And in two other setups, we immobilized the IgG antibodies by a capture chip and used the S1 as analyte here in this setup. And we can nicely see here that the affinities are in a similar range. So we are in a single-digit nanomolar range for Core 101 and a little bit higher for the other candidates of the top three antibodies here. This could also be, or this was also be checked by as a LSA in an independent lab here using the Corat system. So in this setup here, we got also where we were also able to obtain here single-digit nanomolar affinities for the RBD and let's say low double-digit nanomolar affinities for the S1, S2 proteins. Same technology, so LSA was used to identify compatibility groups or epitope bins. So here the antibodies were immobilized on a surface, on a chip, and a pre-mixed mixture of the test antibodies and the S1 and or S2 has been added, and the increase of the signal has been analyzed here by these LSA based system, and we were able to identify three different compatibility groups, which represent in general three different epitope bins where our antibody bind to. After conversion into the Fab fragment, we also co-crystallized the Core 101 antibody with the RBD, and we obtained a structure with a resolution limit of 2 angstrom. And on the bottom in the middle, you see with C depicted here the surface contacts of the RBD to the antibodies D90 C11 and ACE2. And in ACE2, for ACE2, you can see here highlighted the contact region in red. And for Core 101, you see the contact region in yellow. And you see here that Core 101 has a very huge contact region. And in the table on the left, you also see here that the contact region is more than 1,100 square angstrom, which is quite high compared to other antibodies where these data have been available in the PDB databases here. And if you take a look at the structure, you can clearly see here that in the overlay that Core 101 nicely is able to displace ACE2 on the RBD molecule. Next step to check if those candidates can be of therapeutic value is, of course, off-target binding. This has been checked by on cryo sections by tissue cross-reactivity analysis on 34 different cryo sections, and we could not see any misbehavior, any background staining here of COR101 on any of these tissues. Whereas here on the right side, you see a cross-reactive map where you can see here these small brown dots, which refer to, let's say, binding of the antibody to some structure here. On the lung tissue, on the left side, you see the negative control, which looks similar to the staining of COR101. We discussed a lot the format of our therapeutic molecule. And you might be aware that there are a lot of discussions about antibody-dependent enhancement which could occur also for SARS-CoV-2. It's depicted for several viruses such as dengue. There are some hints for SARS-CoV-1 that this might occur. And this antibody-dependent enhancement is an enhancement of infectivity due to the fact that the antibodies bind to Fc receptors and therefore are getting in touch with cells of the immune system, and then these cells might be also infected by those viruses, and therefore, or as we want to, let's say, de-risk the whole molecule, our molecule was, or we concluded that we want to go for a silenced Fc format, meaning an Fc which does not have any binding, or which doesn't show any binding to the Fc gamma receptors, so to avoid any effector function here. And we checked this by biolayer, not by biolayer interferometry, but by surface plasmon resonance using BioCore, and this can be seen here. So you see on the right side here the control rituximab and the affinities to the different Fc gamma receptors, and you see here also that COR101 does not bind to the different Fc gamma receptors, whereas the binding to the neonatal Fc receptor was unchanged compared to the control antibody. Therefore, we pushed all top three antibodies into the CMC assessment. So to check here purity and homogeneity after affinity chromatography, after preparative SEC, we checked thermal stability of the molecule, solubility, viscosity, and stability, in different stability settings here, and we were not able to detect any, let's say, developable or predicted developability issues here. So all three candidates have a very excellent developability profile, and therefore we had to somehow come to the conclusion with which antibody we want to go on into the clinical phase, let's say, into the next steps. And to sum this up here and also give you an idea about the timelines. So in March, beginning March, we entered here the project with different libraries, so with patient-derived libraries and with universal libraries. In May, June, we already had the first live virus-neutralizing antibodies identified, roughly 50 identified here. In the meantime, we established Corat Therapeutics as, let's say, the company which is developing this antibody. Lead detection took place, as I just described here, based on virus neutralization, bioinformatics, and a preliminary CMC Assessment, the top 3 candidates have been used for tissue cross-reactivity and more detailed CMC assessment, and this led mid of July to the final lead, which has then been used for tissue cross-reactivity, so for GMP tissue cross-reactivity, for also in CMC development assessment, and also for tox studies there. So we generated CHO pools of this antibody, and wanted to check if this antibody has the same properties, or if the properties we have seen, let's say, during the discovery phase have been maintained. Therefore, we checked different batches of the molecule for binding to the S1 protein. So we checked Hec material, which has been used for most of the analysis before. We checked a pool, so let's say the pool which have been used afterwards for the GMP production. We used the reference batch and from the same pool, and we used a GMP batch based on a 200-liter scale. And you can clearly see here that the EC50, the binding to the S1 protein or S1 protein, did not differ, and we could also see a slightly increased binding EC50 of the CHO material compared to the Hec material. So far, we only checked virus neutralization in vitro, and we were also able to check, or we also wanted to check in vivo efficacy. This has been done in 2 different models. The first model we used was a Syrian hamster infection model. So Syrian hamsters, they contain an ACE2 homolog to human beings. They elicit a mild to moderate disease, which is associated with pneumonia and a temporary weight loss. And usually they recover very fast after 5 days. And after 14 days, they are usually virus-free. So the setup was that the virus was given intranasally, and 2 hours after infection, the antibodies or the antibody has been given IP, and we used different concentrations here. So we used a concentration of 3.7 milligram per kilogram and 37 milligram per kilogram, which correspond to a human equivalent dose of 0.5 and 5 milligram per kilogram. And we could definitely see a therapeutic effect here. So in yellow, you see the high dose treatment, so 5 milligram per kilogram human equivalent dose, which is 37 milligram per kilogram in the hamster model. And you see here that the weight loss is compared to the low dose model, and also to the control, is let's say it's not that high, and the weight increases much faster compared to the control group. Additionally, we measured the virus titer in the lung in the 3 groups, and we have been, or we were able to see that more than 99% of the virus load has been depleted 5 days after infection in the high dose group. We also checked for histopathological COVID-19 systems, and I mentioned already pneumonia is one of the let's say symptoms of the Syrian hamster model. And you see here that many of these pneumonia symptoms were, let's say, reduced in the treated group compared to the control group. And we also checked for other symptoms, histopathological symptoms, and we could also see here that several other symptoms or COVID-19 symptoms were significantly reduced in the treated animals compared to the non-treated animals. We also used a second in vivo model, which is the ACE2 mice. It's a transgenic mouse model where human ACE2 was transfected into the mice under the control of a keratin 18 promoter for the epithelial expression. And these mice usually get a severe and up to fatal COVID-19 model. And the infectious dose has been adjusted, intranasal adjusted, so that the mice do not lose more than 10% body weight within 5 days of infection in a pretest. Here we used 4 different concentrations with 6, 30, 60, and 100 milligram per kilogram, which correspond to human equivalent dose of 0.4, 2.5, 5, and 10 milligram per kilogram. And these are very preliminary and very hot results because we just were able to have 3 animals per group, but we are currently performing, let's say, more analyses with larger groups here. But you can also see here, compared to the control, so to the untreated control here in red, where we have a clear decrease in weight starting with day 3 post-infection. You see here a clear, let's say, beneficial effect on the weight in the treated groups, already starting with the lowest concentration of 6 milligram per kilogram. The histology is ongoing, and I mentioned already that the groups, so the animal groups, will also be increased in the next experiments here. So to give you an overview about the early development of the molecule, so the process, so the CMC process has been established and was ready in October 2020. The GMP batch, including downstream processing, was done in 200 liter scale and was finished in December 2020. The first proof of concept studies in animals, so in the hamster model, have also been performed in December 2020, and the mice models are still ongoing, but we have already some preliminary data I've shown already to you. PK and safety study, as well as GMP tissue cross-reactivity studies, have been finished in November 2020 without any obvious, let's say, observations there. And IMPD is submitted or was submitted in February 2021, so a few weeks ago. But this is, of course, not the end of the whole story. We all are aware that the different mutations are arising, and it's really a race between, let's say, the therapeutics, the vaccines, and the virus. And therefore, we are checking all the different, or at least major new lines which occur here. And here's just as one explanation here, the Danish mink variant, which has here at position 453 an exchange, let's say, mutation. And you can see here on the right side that, of course, this variant is able to bind to ACE2, but you can also see that Core 101 also binds to this Danish mink variant. We also have investigated already the UK variant. I'm not showing the data here because they are very, very hot and arrived only, let's say, a few hours ago. And we could see also binding to the UK variant. And we also induced mutations in our, or let's say, in the S1 variant during treatment with Core 101, and used NGS analysis to check for escape mutations. And we could also see no enrichment of the 501 mutation in these analyses, which clearly indicates that we also have no problem with the, let's say, currently emerging UK variant. So here is an overview. This will be the last slide. I'm showing to you an overview about the whole development of this molecule here. It's, from our point of view, really fast track. So up to now, we are, let's say, roughly 11 months working on the field. We are hoping to enter the clinical phase 1b in the next few weeks. And we are currently heavily, let's say, working on the clinical development. So for organizing everything for the later stages here. Last but not least, I would like to thank all the people which have been involved in this project, not only the Corat people, not only the UMAP people, also the people from the Technische University here in Braunschweig, and of course all the people which I cannot name here, but which contributed to this project in a very, very valuable manner. And I'm pretty sure without their help, we would definitely not be here at this stage here where we are right now. And thank you for listening to the audience. And now I'm happy to take all your questions.
Well, thank you very much. That was really an inspiring presentation, so much information, and indeed ultra fast development of the antibody therapeutic. So we definitely have some questions coming in. I'll start at the top because those probably relate more to the information you were just giving, and we'll work our way backwards. First, thank you for your work and the very interesting presentation. This particular attendee was wondering, during the library preparation, did you join VH and VL from the single plasma cells or the SCFVs, were these randomly joined VH and VLs?
The VH and VLs have been joined randomly. The reason for that is that we did not, include a paired library, so with combined heavy chain, light chain derived from one B cell, is that the number of B cell clones is limited, and therefore we should be able to cover also the heavy chain, light chain pairing here in library sizes of 10 to the power of 8 to 10 to the power of 9. Maybe not all of them, but definitely most of them.
Thank you. Another question, did your immune library or the naive library or a combination of both work best?
We used both libraries here, but the immune libraries took the lead because they showed in the initial analysis the better neutralization assay, the efficacy.
Next question, did you use CHO cell line or cell pool to produce the GMP batch? If a cell line was used, how did you proceed to GMP so fast?
Yeah, good question. Actually, this is the reason why we used a GMP pool. So we went for a pool and are currently preparing everything, let's say, or we have already the cell line available. But for the GMP pool, for the GMP material, which will be used in the clinical phase 1, we use the pool.
Next question has to do with your plans. Are you filing an IND in the US?
It's not planned so far. We are very, very, let's say, very, very small team. And we are happy and lucky that we got everything in place here for the application for the German authorities. So far, it's not planned, but we do not know, maybe in the future.
Yeah, quick follow-up question on that then. I mean, so you you filed with the Paul-Ehrlich Institute in Germany, then was there also a requirement, or is there a requirement to file a document with EMA?
Sorry, I
the
didn't get
European Medicines Agency, were there also documents filed with them as opposed to just within Germany?
No, we're just going, so far we are just going for Germany. So
okay, good to know. That's a good procedural question that I was interested in from the regulatory aspect.
Yeah, it would be nice, but you know it by yourself. So to file those things, let's say the documents, it's a mess of work. Therefore, we in the first step, we went for the Paul-Ehrlich Institute here in Germany. We also had some scientific advices too during the whole development here to support us also with the idea going with a pool and so on. So and I think it would be a mess to also talk with the FDA in parallel, with the EMA in parallel. So this is something which a small group, let's say, of people cannot handle.
So if you're Roche and you have a huge regulatory affairs department, maybe something else. Another question, did you test neutralization activity against the N501Y mutant?
This is ongoing. We are currently organizing, or at least we have organized the strain. It's already there in our partner labs here at the Helmholtz Center of Infectious Diseases. They are currently preparing the virus stocks, and then we will definitely also test virus neutralization on this variant.
Next question. So did you consider going with a mixed pool? So I think here a combination strategy is what's being asked here. Did you consider going with a mixed pool of antibodies and not a single antibody?
Yes. So this is also under discussion, and this is also a strategy we are going, or we at least trying to go in the next step, let's say, for next phase. But with limited time and limited resources, we were definitely we would definitely not at this stage here if we would have gone with a pool of 2 or even 3 or more antibodies. This would mean a lot of more work, a lot of more resources, and we wanted to really have a fast track approach. Not only to let's say have a product ready as soon as possible, but also to treat people as soon as possible. And this is definitely with limited resources, not an easy task.
Next question. Have you been able to characterize the specific motif that is recognized by Core 101? If it recognizes and neutralizes even the mutant variants, do you think that there may be a conserved sequence that could be explored for vaccine development against future mutants?
Yeah, interesting question. So I doubt that this is possible, because if you take a look at GS8, at least in the RBD, there might be other, let's say, areas of S1, maybe also in the RBD, but I doubt that. But if you have a look at the huge amount of different variants and of different mutants which occurred in just 1 year, without, let's say, there's no huge pressure towards mutations, so far. So that I doubt that there will be such motive, but therefore we did not look for that.
Did you jump directly in pivotal GLP tox, or did you do pre-studies?
We did some pre-studies in a mouse model, and then we switched to the tox study.
Did you find any allosteric inhibitors?
Yeah, it's hard to predict because we do not know all the epitope of all of our antibodies. So it would be nice to have an antibody which binds outside of the RBD and which also is able to block the interaction to ACE2. This might be possible, but we focused solely on the RBD, and all antibodies which were only binding, let's say, or which bound to the outside of the RBD to the S1, they have been, let's say, not been pushed into the next phase because we just moved forward fast track. As I mentioned, you cannot look left, you cannot look right, you just have to go straight forward. It would be very, very nice to have a look on that, to do some biology, to do some science, to be honest, yeah. But this is something we will definitely do in the near future once we have some air to breathe to do these kind of analyses.
Just to double check, I'm not sure you actually came out and specified, but can you say what the dose is for your phase 1 study? And presumably this is an IV administration, so it's formulated for IV administration.
That's true. Our dose, I cannot give you the exact numbers. There will be different concentrations, but our target dose is 10 milligram per kilogram.
Question, going back a little bit further in your presentation, why wasn't solubility of the antibody in full IgG format evaluated early in parallel with activity?
Could have been done. Actually, this would also need some, let's say, some material. Yeah. So these kind of assays need larger amounts of material, and if you take a look here on the timelines, you definitely see that there's no, let's say, no time left to produce larger amounts of a large number of antibodies to do these kind of CMC assessments. So, and therefore, we let's say, did this analysis at a later stage where we already have, let's say, large quantities available from HEC expression, where we could do these kind of analyses. Yeah, it's just a matter of this fast track here. Would be nice, yeah, but it's definitely, as I mentioned, for such a small team, not possible to do it with let's say, tens or hundreds of antibodies.
Since you're using pool material, does the antibody show variation or variety during characterization?
We did not. We checked that, of course, but we did not see any variation or similar things, because this would be a major issue where I think the regulatory authorities would have a look on that.
Next question. So in a therapeutic setting, says there seems to be a low risk for Cora 101 as you're binding to the virus and removing it from the situation. But what about in a prophylactic setting? Which information would Cora 101 trap that normally would find its way to the ACE2 receptor and is then missing?
We do not go for, we do not develop for prophylactic treatment here. First of all, it's a totally different setup in the clinics. You can imagine if you're doing a prophylactic setup, you have to treat hundreds and thousands of people to fulfill the statistic needs. And this is also more or less impossible for a small company. Yeah? And second of all, we would definitely not go for for silenced FC, which is cleared, let's say, half-life of 2 to 4 weeks. We would have gone for an FC enhanced version to prolong the half-life in the serum. So therefore, we do not develop for prophylactic treatment. It would be possible, but this is not intended here.
Okay, for last question or so, so the title of your presentation is about this ultra-fast development. So this is more of your opinion. of the technologies that you used or ones that others are using, what do you feel or what do you consider that are the key technologies that allowed you to do this rapid development?
And to be honest, it was a team. So we had a lot of support from different teams here all over Germany. So I mentioned already some of them. So the Helmholtz Center of Infectious Diseases, who established very, very fast, within a few weeks, the SARS-CoV-2 infection model on virus-6 cells here to enable us to screen in a very fast in a very fast approach for neutralizing antibodies. Otherwise, we would have worked with surrogate antibodies or pseudoviruses or similar things. And we definitely know also that pseudoviruses, they're not the same than live virus isolates. So then we had great support from the Technische University in Braunschweig. I mentioned already, we had great support from the Universitätsklinikum in Tübingen here in Braunschweig, who helped us a lot with, let's say, some clinical input at this stage. We have great support from another group here in Germany who helped us with all the CMC development, CMC assessment, cell line development, and so on. So I think it is a team because we are not Roche. Yeah, you mentioned it already, Jan. We are just a small company with limited resources, and if you have this very strong background scientifically and also let's say supporting us in other ways, yeah, then I think this is a key to success, how we were able to make it that possible.
Well, thank you very much. We are at the top of the hour, so in concluding, I would definitely like to thank Dr. Frenzel for relating his fascinating experience with this incredibly fast development of an anti-SARS-CoV-2 antibody, and I definitely wish Corat Therapeutics much success. I thank you for attending the webinar today. An on-demand version will be available in a few days. I'll send a link to it by email to everybody who registered. And so please do feel free to watch this or any of our other on-demand webinars when it's convenient. With that, thanks again and have a great rest of your day.